Hi all, welcome to Adihuda second level online classes. Today we are going to study the differences between simple microscope and compound microscope. We already studied what are the differences between simple microscope and compound microscope. The eminent famous scientist, the Robert Hooke, who discovered the structure of the cell by using microscope. Later, you can see there are many big advancements in medical field, microbiology, all this. So we can identify the microscopic object by using microscope. Nowadays, we are facing the threat of coronavirus. We can identify these microbes, unicellular organisms and microcellular organisms. The cell structure, the cells we can identify by using microscope. So, which are the examples of microscope? Simple microscope, compound microscope, electronic microscope. Different varieties of microscopes are there. Here, from these, we are mainly using two types of the microscopes, that is compound microscope. We are using compound microscope. In school labs, you can see compound microscope. You can see electronic microscope. Big laboratories, they're using electronic microscope for the better, clear understanding of the microscopic object. We will get the clear object, clear image when we use electronic microscope. When we use compound microscope, we not get that much bigger. What are the difference between compound microscope and electronic microscope? We can look compound microscope. The structure of compound microscope, that's very familiar to you all. What are the difference? One is that magnifies object 2000 times. Compound microscope can magnify when we observe any object by using microscope. We can see that object 2000 times magnified. That much bigger. It can magnify the object 2000 times. Here, which is a source? Here we are using light source. Light is the source of compound microscope. Light is the source of compound microscope. By using light, you can see the adjustment. Condenser, just below you can see the structure of mirror. By using this mirror, we can adjust the structure, the light source, the natural light from the sunlight that we are using here. So we can say light is a source for this. Okay. And stage, clip, everything you can see in compound microscope. That's very familiar. Magnify 2000 times. These are the two important points. Whereas electronic microscope, that's not familiar to you all, because the electronic microscope mainly used, you can see in laboratories and big labs. Here, we can magnify the object about million times. Approximately million times clearly. We can magnify the object million times clearly. That is the importance of electronic microscope. Very much bigger. Okay. And electrons are using here in electronic microscope. Whereas in compound microscope, light is using. Okay. These are the important difference between compound microscope and electronic microscope. This a question you can expect in your question paper, that what are the difference between electronic microscopes and compound microscope. This is the difference, magnify 2000 times and light is the source. Electronic microscope magnify the object million times clearly. The source is that electrons are using. Get me? I think you all got me. Okay, When you have any doubt, you can ask. These are the difference between compound microscope and electronic microscope. Okay, next we can study the difference between cell and the nucleus. The structure of nucleus is very familiar to all. Okay, students, next we can study the structure of a nucleus. The structure of a nucleus is very important. When we discussing in detail, when we discuss the structure of a nucleus, outer cell you can see the cell structure, inside you can see center is nucleus. The structure of nucleus is very important when we discussed. What are the important stages or which are the important parts of a nucleus that we can discuss? This is the structure of a nucleus. The simplified form is this. You can look the picture, correct picture I am giving here in this video. So this is the structure of a nucleus. In this picture, you can see different parts of a nucleus. First you can see nuclear membrane. Nuclear membrane. This is the nuclear membrane. Nuclear membrane. Did you get me? This is the nuclear membrane. Then this one is nucleoplasm. This fluid part is called nucleoplasm. The outer membrane is called nuclear membrane. This one is nuclear membrane. The fluid part is called nucleoplasm. Very simple. And the center of nucleus. Here you can see nucleolus. This one is nucleolus. Nucleolus. So which are the important parts? Nuclear membrane, nucleoplasm, nucleolus. Inside the nucleolus, you can see chromatin reticulum. Chromatin reticulum. These are the important parts of nucleus. Nucleus seen inside the cell. At the center, nucleus is the center of the cell. Nucleus is the center of the cell. Okay. The center of nucleus is that nucleolus. Nucleolus is the center of nucleus. Nucleus, its center is nucleus. Nucleolus, not nucleus. Nucleus center is called nucleolus. But center of cell is called nucleus. This is the difference. Okay. Did you understood what I am saying here? I hope you are understand. Okay. This is the cell. This is a nucleus. The center is called a nucleolus. The outer membrane is called a nuclear membrane. Nuclear membrane. The inner part is called, this fluid part is called nucleoplasm. The picture you want to send me. Everyone draw this picture and send to me. Okay, personally. I will check your notebook. Then inside the nucleolus, you can see chromatin reticulum. What you can say? Chromatin reticulum. You can say along with me. These are the important parts of a nucleus. Okay. Then when we discuss in detail the structure of a nucleus, nuclear membrane. What is nuclear membrane? Double layered. Nuclear membrane is the double layered membrane that cover the nucleus. Nuclear membrane, that is a membrane. Nuclear membrane is the membrane, double layered membrane that cover the nucleus. That's why it's called a nuclear membrane. Double layered membrane that cover the nucleus. That is nuclear membrane. Okay. Second one is that. So, which is a covering of this nucleus? That is nuclear membrane. Then the fluid part of the nucleus is called nucleoplasm. Nucleoplasm, like cytoplasm. The fluid part of a cell is called cytoplasm. The fluid part of nucleus is called nucleoplasm. The fluid part, free space of the nucleus is called nucleoplasm. Next we can look nuclear pore. Very simple. Nuclear pore help to, this is a minute opening. Nuclear pores are the minute opening that's seen on the surface of nucleus, nuclear membrane. Small opening you can see on the surface of nuclear membrane. That helping for conduction of materials. Conduction of materials to in and out. In and out. That help to conduction of materials in and out. this opening, small opening, is called nuclear pore. That nuclear pore helping for the conduction of materials in and out. Okay, next we can discuss chromatin reticulum. Chromatin reticulum, you can see a network, you can see in nucleus, in nucleolus. A network seen in the nucleolus. Okay, nucleus, you know, the center portion that synthesizes proteins. Okay, at the center portion is called a nucleolus. There inside the nucleolus, chromatin reticulums are seen. What is chromatin reticulum? You can see a network like structure. That structure is called a conduction of that, you can see network like structure. That structure is called actually chromatin reticulum. That structure is called this networks like structure is called the chromatin reticulum. Later this chromatin reticulum condensed to form chromosomes. You know that chromosomes, structure of chromosome. So how chromosomes are forming? Chromatin reticulum condensed to form chromosomes. That chromosomes carrying, or this chromatin reticulum carrying genes from parents to offsprings. Sometimes many features or many peculiarities seen, many characters are seen us that are very similar to our parents, mothers like, of fathers like. Maybe you all like maybe your father or like maybe your mother. These are the because this that is due to the presence of genes. Okay, that where we can see the genes, the presence of genes in chromatin reticulum. Okay, this is the structure of a nucleus. This one is very important. Structure of nucleus, I am concluding once again. Okay, this is the nucleus. Nucleus outer membrane is called a nuclear membrane. Then fluid part is called a nucleoplasm. Nucleoplasm. Next, this opening, you can say there is a minute openings are there. That's helping for the conduction of materials. That is called a nuclear pore. Okay, the center is called a nucleolus. Inside the nucleolus, we can see chromatin reticulum, like chromatin reticulum, how in the form of a network that carrying genes. Okay. Dear students, the structure of nucleus. When we observe it closely, you can see different parts. We already discussed nuclear membrane, chromatin reticulum, nucleoplasm, nucleolus, nuclear pore. These are the important parts of a nucleus. Here, nuclear membrane. It is a double-layered membrane that covers the nucleus. Nuclear membrane is the double-layered membrane that covers the nucleus. You can observe this structure of nucleus very closely here. Second portion is that nucleoplasm. Nucleoplasm, it is a fluid part of a nucleus. Nucleolus and chromatin reticulums are seen here. You can see there nucleoplasm. Nucleolus is that there are spherical bodies. Nucleolus is the spherical body that play a major role in the synthesis of ribosome that we already studied. Next, you can look nuclear pore on the surface of a nuclear membrane. Nuclear the last part is that in the nuclear membrane, they help in the conduction of materials to and from the nucleus. Gene. Genes are carried by chromatin reticulum. Okay, these are the important parts of nucleus. Okay, nuclear membrane, nucleoplasm, nuclear pore, nucleolus, chromatin reticulum. Okay, dear students, next thing we can see a special cell organelles seen in plants. That cell organelle is called plastids. What is plastids? Plastids are the cell organelles that are exclusively seen. Plastids are the cell organelles that are exclusively seen only in plant cell. Okay. What is plastid? Plastids are the cell organelles that are exclusively seen in plant cell. They are called plastids. Only seen in plant cell. plastids examples is that you can see mainly three types of plastids are there. Which are the examples of plastids we can discuss now. That is very important. What is plastids? Plastids are the cell organelles. Similar like chromatin reticulum, ribosome, Golgi bodies, mitochondria, or like this. Plastids are cell organelles that are exclusively seen in plant cell. They are seen in plant cell. Which are the examples of plastids? One you can say chromoplast. First one is chromoplast. Second one is chloroplast. Third one is leukoplast. You can say chromoplast. Did you get me? Chloroplast, chromoplast, leukoplast. First one chromo, chloro, leuko. Three types of the plastids are there. Chromo, chloro, leuko. Chromoplast, chloroplast, leukoplast. Chromoplast, chloroplast, leukoplast. These three are plastids. Examples of plastids. Three types of plastids are there. One is chromoplast, second one is chloroplast, third one is leukoplast. They are performing different functions. What are the different functions done by these plastids? First, chromoplast. We can discuss the structure of the chromoplast function. Impart color to flower and leaf. Chromoplast providing color to leaves and fruits, flowers, everything. The presence of chromoplast. You already studied the examples of chromoplast. You already studied from lower classes. Okay, which are the examples of chromoplast? They are providing colors to the leaf. When we take a flower, the color of flower become red. It's a red color flower you can see. When we take any other leaves, you can see colorful leaves are there. That is due to the presence of chromoplast, not chloroplast, not leukoplast. This chromoplast impart or provide color to the flowers and leaf. Which are the example? Xanthophyll, carotene, anthocyanin. Xanthophyll, carotene, anthocyanin. Carotene, you already studied. These are the examples of chromoplast. Chromoplast is a plastid. Okay. What is the function of xanthophyll? What is the color of xanthophyll? Yellowish color. Yellow color. You can see many yellow colored flowers. That is due to the presence of xanthophyll. Xanthophyll is a chromoplast. Chromoplast is a plastid. Okay. Next, carotene. Carotene, yellowish orange color. Carotene, yellowish orange color. Anthocyanin, red purple color. Red or purple color. Anthocyanin. Can you say which are the examples of chromoplast? Anthocyanin, carotene, xanthophyll. All these are the examples of plastids, that chromoplast. Then chloroplast. Actually, chloroplast, what is the changes? The chloroplast, before I want to discuss leukoplast. We can discuss leukoplast. Then we will discuss then leukoplast. The chloroplast, okay, look, you can see here chromoplast and chloroplast and leukoplast. The chloroplast will changes to chromoplast. The chloroplast will changes to chromoplast when fruit ripen. When fruit ripen, the chloroplast will changes to chromoplast. The color you can see, the ripened fruit colors you can see. Then starch is converted into sugar. That's why we will get very sweetness. Okay, the mango. Ripened mango, when we taste it, we will get sweet. We will feel that. Okay. Then what is the importance of leukoplast? Leukoplasts are seen where the food materials are stored. There you can see leukoplast. Food materials are stored. There you can see leukoplast. These are the important plastids seen in plants body, that are chromoplast, chloroplast, leukoplast. Chromoplast impart color, but plastids are the cell organelles that are exclusively found in plant cell. They are found three kinds we already studied: chloroplast, chromoplast, and leukoplast. Okay. Each plastid is performing different functions. Chloroplast changes to chromoplast when fruit ripen. Then leukoplast, we can see the food materials are stored in plants. Okay. Let me conclude my class. Thank you.